Hi guys, my name is Jason Matthew, and today we're going to be looking at the cell, which is normally dealt with in the level 1 courses at the university. Now if you haven't done so as yet, please join us on Facebook. Do a search for Biochem GM. We would love to hear your comments. Please let us know how we can improve this service to better enhance your learning experience. Also, if you have any materials, slides, figures, diagrams that you think would be helpful, please email us jasonmatthew2011@gmail.com. So today's lecture, at the end of this, you should be able to do a few things. You're supposed to be able to discuss factors that affect cell size, compare eukaryotic with prokaryotic cells, compare plant and animal cells. Describe the structure and function of the following organelles. And these are the list of the organelles that we're going to be describing the structure and functions of in the subsequent slides. Alright, so Robert Hooke discovered and described the fundamental unit of all living things, and he called it the cell. So what is exactly a cell? A cell is the smallest unit that is capable of performing life functions, which are growth, metabolism, stimulus, and replication. Now all these are examples of cells, and as you can see, they differ in shapes and sizes, and that's because they have different functions. So how small can we go? Well, the bacteria, which is the smallest free-living cell, measures 0.5 by 0.5 by 1.6 micrometers. And any cell smaller than a bacterium is unlikely to have sufficient DNA to make the enzymes necessary to catalyze the biochemical reactions that make independent life possible. So this sets a lower limit on the size of a cell. Now, if you do some other reading, you might see that viruses are actually smaller than the bacteria. So for some homework now, what you should do is go and see what mechanisms they have in place that is different from bacteria that will allow them to be smaller. All right, is it that the DNA is smaller, or they have less DNA? So please go and check it out. All right, one more thing here, you have this here, which is micro. Now micro stands for 1 by 10 to the minus 6, or in other words, 1 millionth. So why can't organisms be one big giant cell? So in other words, we're looking at the other end of the spectrum. We just looked and saw that cells could only, there's a certain limit of how small a cell can go, and that limit is set on the amount of DNA that is needed to make the enzymes necessary for life. Now on the other hand, cells can't just get infinitely big. There is a limit. Alright? And this is the limit, the reason for it. Most cells are relatively small because as size increases, volume increases much more rapidly. So there's a longer diffusion time. And because of that, because it's going to take longer for substances to diffuse in and out of the cell, that would have a limitation on how big a cell can get. Now let's look at the concept of surface area to volume ratio. Because when you all are discussing how big a cell can get, you need to do it in reference to the surface area to volume ratio. So here we have 3 cubes. Alright, and obviously they are increasing in size. Now the surface area for this 1 millimeter cube will be, well, the area of one square, or one side, which is 1 by 1 squared. And there are 6 sides, so it's 6 millimeters squared. And similarly, you find the surface area for this cube and this cube. And you see it went from 6 to 24 to 96. Alright, so that's how the surface area is increasing. But look at the volume. The volume is going from 1 millimeter cube to 8 millimeter cube to 64 millimeter cube. So we see that the volume is increasing much more rapidly than the surface area. And when we divide the two to find the surface area to volume ratio, we see that although the cubes are increasing in size, when we look at the surface area to volume ratio, it is decreasing. It goes from 6 to 3 to 1.5. So there's less surface area to volume ratio. And that in essence is what limits how big a cell can get. So as we see here, volume increases more rapidly than surface area. And if you look at the example before, the cell size doubles. As the cell size doubles, there's 8 times as much volume, but only 4 times as much surface area. So in conclusion, as cell size increases, the ratio of surface area to volume decreases. Therefore, the rate of diffusion slows down. Thus, a cell cannot get rid of waste fast enough or get nutrients fast enough. So it will either be poisoned by its waste or starve. All living cells fall into one or two broad categories, prokaryotic and eukaryotic. The distinction is based on whether or not the cell has a nucleus. Prokaryotes are single-cell organisms that lack nuclei and other organelles. The word is derived from pro, meaning prior to, and karyote, meaning nucleus. So prokaryote means prior to nucleus. So prokaryotes have no nuclei. Nuclei being the plural for nucleus. Now, look at this. Prokaryotes are single-cell organisms. So all prokaryotes are single-cell organisms. But please don't confuse that with saying all cell organisms are prokaryotes. That is not right. So I'll just repeat that. Prokaryotes, all prokaryotes are single-cell organisms. But all single-cell organisms are not prokaryotes. So in conventional biological classification schemes, prokaryotes are grouped together as members of the kingdom Monera, represented by bacteria and cyanobacteria. The other 4 living kingdoms are all eukaryotes, the single-cell protists such as amoeba, and all multicellular life forms, including the fungi, plant, and animal kingdoms. So remember I was saying that not all single-cell organisms are prokaryotes because, for example, amoeba, which is single-cell, is a eukaryote. Now, eukaryote cells have true nuclei and other organelles such as mitochondria. And the prefix "eu" means true. And if you remember from the slide before, karyote means nucleus. So they have a true nucleus. So just to recap, prokaryotes have no nucleus. Eukaryotes have a nucleus. So common prokaryotic structures. You'll have a cytoplasm, genetic material, cell membrane and cell wall, ribosomes, and some will have flagellum to move around. But all prokaryotes will have no membrane-bound organelles. Membrane-bound organelles are found only in eukaryotic cells. So here we're looking at a typical or general prokaryotic cell. And we see that on this side will represent the structures that are always found. And you see that in a prokaryotic cell, there's a cell wall. Alright, now the cell wall is composed of a different material from the cell wall as in plant cells. Alright, now you should know by now that plant cell walls is composed of a material called cellulose. There is no cellulose in the cell walls of a prokaryotic cell. It's made up of something else. Alright? So I won't give you that answer. I'll let you do some research and see what is the main material, alright, that makes up the cell wall of prokaryotic cells. They have a cell surface membrane, cytoplasm, ribosome, and circular DNA. So this is circular DNA. And we'll talk a little bit more about that just now. Now, in additional structures that are sometimes present, you have pili for attachment to other surfaces or other cells. It's involved in sexual reproduction. You have the plasmid, which is a small circle of DNA, right? And if you go ahead, you would see that plasmids play a big role in resistance and so on. Mesosome is an enfolding of cell surface membrane associated with DNA during cell division and helps with formation of new cell walls. Have a capsule for additional protection and a flagellum for mobility. So this is another diagram just to reinforce the common structures found in the prokaryotic cell. Now, a term that wasn't in the previous diagram was nucleoid. Right? So the nucleoid is what contains a single, simple, long, circular DNA molecule. Key thing here being it's simple and it's circular DNA molecule, and we call that the nucleoid. Alright? Now, the only organelles that the bacteria really have would be ribosomes. And you will also see that in the eukaryotic cell, there are also ribosomes present. But the ribosomes in bacteria are much smaller, and they are 70S, while the ribosomes in eukaryotic cells are 80S. Now, while bacteria normally gets a very bad rap, we always think of bacteria in terms of causing disease, and we have to take antibiotics and so on. But there's also useful bacteria, and I just thought I should mention that. Alright, so some bacteria can degrade oil used to clean up oil spills. Others uses for bacteria include making yogurt, cheese, and buttermilk. So there are some very useful bacteria out there. Not all are bad. Alright, so differences between prokaryotic and eukaryotic cells. Now, prokaryotic cells tend to be much smaller than eukaryotic cells, and they are around 0.5 micrometers as opposed to 40 micrometers in eukaryotic cells. The prokaryotic DNA, it's circular and it's naked. In other words, there's no housing for the DNA. It's just naked there in the cytoplasm. For the eukaryotes now, the DNA is in the form of linear chromosomes, and they are found in the nucleus. Alright, so the nucleus is an organelle that houses the DNA for the eukaryotic cells. Alright, now the DNA itself in the eukaryotic cells are much more complex than the DNA in prokaryotes. And we see that there are also proteins called histones that are associated with the DNA molecule in eukaryotes. Now, in terms of organelles, there's only one organelle that the prokaryotic cells have, and that will be ribosomes. For the eukaryotic cells, they have many organelles. Some are double membrane, such as the nucleus, mitochondria, and chloroplast, while some are single membrane, example the Golgi apparatus, endoplasmic reticulum, and lysosomes. A point that should be also made is that prokaryotic cells, they have no membrane-bound organelles, meaning that whatever organelle that they have there, it does not have a cell membrane. Alright? Well, a membrane, a lipid bilayer. So if you put the links together, you know that ribosomes do not have a membrane. Now, prokaryotic cells, they have a cell wall, and it's made up of glycoproteins, mainly murine. Alright? For the eukaryotic cells, not all eukaryotic cells will have a cell wall. So let's recap. All prokaryotic cells will have a cell wall, but not all eukaryotic cells will have a cell wall. For instance, animal cells have no cell walls. Alright? But plant, they have a cell wall, but it's made up of a different type of material. Plant cell walls are made up of cellulose. Alright? Fungi also has a cell wall, and that cell wall is mostly made up of chitin. And the final difference that we're going to be looking at in this table will be the ribosomes. Yes, both prokaryotic and eukaryotic cells contain ribosomes, but the prokaryotic ribosomes are slightly smaller, and they are 70S, while eukaryotic ribosomes are 80S. So these are some of the cell organelles that we're going to be looking at. So we have the nucleus, all right. We have the cytoskeleton, the mitochondria, the chloroplast, all right, and so on. All right, so this here is a typical cell, and you know we could link some names to these different organelles according to their function. So let's start with what we could say is the biggest or largest organelle in the cell, which is the nucleus. All right, it's easily seen with a proper light microscope. All right, and it's called the control center. And the reason why it's called the control center is because inside of the nucleus contains the DNA or the genetic material that controls the activity of the cell, simply because it has the genes that would code for all the enzymes and proteins necessary for all the functions of the cell. All right, it holds the blueprint, in other words. Now, inside of the nucleus, there's a densely pigmented area called the nucleolus. And inside of the nucleus, its function is to manufacture ribosomal RNA. So inside of the nucleolus, ribosomes are made, an RNA, ribosomal RNA. So that's the nucleus. If we go to the cytoskeleton now all throughout the eukaryotic cell, there's a network of microtubules that is called the cytoskeleton. And the cytoskeleton is sometimes described as the scaffolding of the cell. And basically it gives the cell its shape. Alright? As well as it's like it's used to move around the organelles back and forth in the cell. The other one we have here is the mitochondria. And the mitochondria is described as the powerhouse of the cell, because this is where aerobic respiration will take place. And you'll be manufacturing ATP. Alright, ATP will be the energy currency of the cell. So you're going to be making energy or producing energy in the form of ATP. Now we have here the lysosomes. Now the lysosomes are found only in animal cells, and they are what we call the cleanup crew. Alright? And basically in this lysosome there are different enzymes that will be used to break down different materials. Like for instance, they have proteases, and proteases will be enzymes that will break down proteins. They have lipases, which will break down lipids, and so on. Alright? So the lysosomes is the cleanup crew, and it's found in animal cells. Let's see. Now we go here to the Golgi complex or the Golgi apparatus. And you see here that the Golgi apparatus is what we call the distribution center, or in other words, the FedEx, or if you're in Trinidad, the TT Post. In other words, what happens is that proteins such as enzymes and so on that is synthesized by the ribosomes in the rough ER, they are sent to the Golgi apparatus. And what the Golgi complex will do then, it will package and basically put an address on where these enzymes, where these proteins are supposed to go. Right? The ribosomes, now ribosomes can either be free in the cytoplasm, or they can be attached to the endoplasmic reticulum. Alright? And they are the site for protein synthesis. Alright? And last but not least, we have the endoplasmic reticulum. And if you watch this diagram carefully, you will see that the nucleus, which is a double membrane structure, so it's made up of two membranes, an inner membrane and an outer membrane. The outer membrane of the nucleus forms a continued folding, which forms the endoplasmic reticulum. Now, some parts of the endoplasmic reticulum, you will see that it will be studded, and it will be studded with ribosomes, and we call that the rough ER. And the ribosomes, as we already saw, they make proteins. There's where protein synthesis takes place. And in the rough ER, the endoplasmic reticulum in the rough ER is responsible for post-translational modification. And what that simply means is that, well, first of all, I don't know if you know what translation is, but translation was when you took the information from the mRNA and you made proteins. Now, that would have taken place on the ribosomes. So what we say post-translational modifications, we mean the simple little modification that takes place after the protein is made on the ribosome. So the rough ER, the endoplasmic reticulum, would do some post-translational modifications such as add a carbohydrate group or put a signal sequence on it. All right, so that's what the ER and the rough ER does. All right, now the proteins that the ribosomes on the rough ER tends to make will be those that will be secreted out of the cells, such as the hormones and digestive enzymes, for instance. All right. Now, ribosomes can be also free, as we said, in the cytoplasm. These ribosomes tend to make proteins that are more intracellular, within the cell. Now, back to the endoplasmic reticulum. As I said, the endoplasmic reticulum, you could either have rough ER, right, and that is where you have ribosomes attached to the endoplasmic reticulum. Or you could also have endoplasmic reticulum that has no ribosomes on them, and we call that smooth ER. Now, smooth ER, there's no protein synthesis taking place because there's no ribosomes present. And ribosomes are where protein synthesis takes place. So the smooth ER does something different. It has nothing to do with protein synthesis. On the smooth ER, we have things like lipid metabolism taking place. You have steroid synthesis, such as the synthesis of cholesterol. And you also have a lot of detoxification of drugs taking place. So the smooth ER is particularly active in like the liver, where a lot of detoxification takes place. All right. So this here is a general schematic of an animal cell. So as I said before, lysosomes, they are present in animal cells. They are not found in plant cells. Another organelle that is found in animal cells but not in plant cells would be centrioles. So you have centrioles and lysosomes that are present in animal cells, but they are not seen in plant cells. All right. So here we have some basic organelles that are present in animal cells, and their functions. And we have discussed most of these. The only one that we didn't say about was peroxisomes. And peroxisomes, they basically have enzymes in them that will destroy any peroxide that is formed. All right. Other than that, I believe we covered everything here. All right. So what this is showing you here is that in this figure here, this is an animal cell seen under the light microscope. And if you watch this diagram, it is the same animal cell, but now you're looking at it via an electron microscope. And you can see definitely there is more resolution with the electron microscope, and you're able to see more. There's more detail in the electron microscope. For instance, one thing that we can't see before when we used to use the light microscope is that we can now see the endoplasmic reticulum as well as the ribosomes. All right. So with the invention of the electron microscope, and when scientists started looking at the cell through the electron microscope, well then the whole way in which we looked at the cell and the way how we understood biology, it changed astronomically. Right? We will no longer be the same when we started to look at the cell through the electron microscope. Everything changed after that. Alright, so ribosomes. The ribosomes are made up of a large subunit and a small subunit. Alright, so ribosomes are RNA protein complexes composed of two subunits that join and attach to the messenger RNA. So basically what happens is that the mRNA there's a binding site on the small subunit for the mRNA. The mRNA will bind the small subunit, alright, during protein synthesis. Alright, so the ribosomes are the site of protein synthesis. And as we said before, ribosomes are made in the nucleoli. And the nucleoli is found inside of the nucleus. Now the role of the Golgi apparatus. Remember what we said before, the Golgi apparatus is the shipping center, like the FedEx or the DHL or whatever, right? And I think I should have a better diagram. Yeah. So let's look at this diagram here. So remember the endoplasmic reticulum is continuous with the outer membrane of the nucleus. And there will be ribosomes on the rough ER, and that will be the site of protein synthesis. Now once the protein is synthesized by the ribosomes, then they are ready. Oh, sorry, come back here. Then they will be ready to be transported to the Golgi apparatus. Now, we say that the Golgi apparatus is often said that the Golgi apparatus is described as like flattened pancakes, They make that association. Now, once the proteins are manufactured and there's a post-transition modification in the rough endoplasmic reticulum, it is then sent to the Golgi apparatus. Now, the Golgi apparatus have a cis phase and a trans phase. Now, in the cis phase is where you'll be accepting transport vesicles from the rough ER. Alright? They will bind with the Golgi apparatus where there will be certain signals will be placed on them and they'll be modified as well, and so that they'll know which proper destination. There'll be all the proteins in the Golgi apparatus will be sorted and then packaged to be sent to the various destinations that they're supposed to go. Now, the trans phase is where things are shipped away, and you form the secretory vesicle and it's then shipped out. Alright? So these are the different steps taking place. Alright, so the job of the lysosomes now. So again, the same process. The lysosome is a single cell organelle that contains digestive enzymes such as lipases, nucleases, proteases. Alright? They will be initially formed by the rough ER. Alright? The ribosomes would have synthesized the enzymes, and then the rough ER would have done some post-translation modifications. And once that has happened, it's then packaged, it's then put in a transport vesicle and sent to the cis phase of the Golgi apparatus. The Golgi apparatus now will sort out all the proteins that the rough ER just sent them, and they will package them. And basically, on the trans side, there'll be vesicles that'll be budding off, that'll be lysosomes. Alright? Now, this lysosome here will be a primary lysosome. And then it will fuse with a vesicle, forming a secondary lysosome. And this is during phagocytosis. So the cell engulfs some material from the outside. It forms a food vesicle, and then the primary lysosome will bind to it, forming a secondary lysosome. And what happens is basically that the contents of the primary lysosome, is released into the vesicle here, and digestion of the phagocytized food particles or cells take place. All right. Now, another organelle that is responsible for protein degradation is the proteasome. Now, why is degradation important? Degradation supplies amino acids for fresh protein synthesis, as well as it removes excess enzymes. There are two major intracellular devices in which damaged or unneeded proteins are broken down. We have the lysosomes, which we discussed in the previous slides, and now the proteasomes. The proteasomes deal primarily with endogenous proteins, that is, proteins that were synthesized within the cell. So if you look at this slide before, you saw that the lysosomes was more responsible for particles that was outside of the cell. All right. Now, the proteasome and the lysosomes, they are responsible for degrading proteins that are within the body. All right. Now, there's also digestion of dietary proteins, which will involve enzymes in the stomach and the pancreas and so on. That is something different, all right? Now, lysosomes, I should just point out that proteasomes are solely involved in protein degradation. Lysosomes are involved in protein degradation, but they also degrade other biological molecules such as lipids and nucleic acids. All right, they are as we saw before, they are the cleanup crew of the cell. All right, peroxisomes, they contain oxidative enzymes that degrade amino acid and fatty acid. Hence, peroxisomes contain amino oxidases, hydroxy acid oxidase, and catalase. The latter enzyme, catalase, is necessary for the breakdown of hydrogen peroxide, which is H2O2, which is a byproduct of other enzyme catalyzed reactions. And H2O2 is necessary to break that down because that could cause a lot of cell damage if left. Alright? Peroxisomal enzymes are synthesized on the free cytosolic ribosomes with a signal sequence that directs them to peroxisomes. Alright, so mitochondria, and that's the powerhouse. Alright? So the mitochondria is made up of several things. It's first of all a double membrane structure. So it has an inner membrane and an outer membrane. Now inside of the mitochondria there is the matrix. Alright? And the function of the mitochondria is to produce ATP. Also inside of the mitochondria there is DNA as well. So there's a point of interest that we know the nucleus contains the DNA of the cell, but that's not the only organelle in the cell that has DNA. We're now seeing that the mitochondria as well has DNA inside of it, and because that's one of the reasons why we think that mitochondria were originally prokaryotic cells. And there's an interesting theory called the endosymbiotic theory that goes into this and explores this possibility. All right. So you see here we have it's made of two membranes, the outer and the inner membrane. The inner membrane is folded to form cristae, and then inside of the mitochondria we have the matrix. The cytoskeleton is made up of several tubules. We have the microtubule, microfilaments, and intermediate filaments. And as I said before, they are what some people describe as the scaffolding of the cell. Right? It's all throughout. Well, this is only for the eukaryotic cells. That is, it's all throughout the eukaryotic cell, and it gives that cell its characteristic shape. All right. Also, the cytoskeleton is used to transport organelles from one part of the cell to another part. So if you see like the mitochondria, it's like it's hooked up to the cytoskeleton, and then the cytoskeleton is going to move it along the cell. Alright, so general schematic of a plant cell. Now, the plant cell will tend to be very similar to what we saw in the animal cell. It's going to have a Golgi apparatus. It's going to have mitochondria, nucleolus, nucleus, and so on, endoplasmic reticulum, ribosomes, and so on. So other additional structures that the plant cell will have that the animal cell didn't will be things like the plasmodesmata, the chloroplast cell wall. Alright, these are some main things that the plant cell will have that the animal cell won't. Alright, so we already spoke about most of these things. Just to point out the chloroplast, that's a double membrane structure. Alright, and it harvests sunlight, produces ATP and carbohydrates. Alright. Plasmodesmata, it provides part between two plant cells. And basically the other structures, we have already dealt with them when we looked at animal cells. The only thing I want to point out here is that the vacuole, while in animal cells, you tend to see some small vacuoles. In the plant cell, you tend to have this one large vacuole that takes up most of the volume of the cell. So that is just something to remember. Alright, so this is the plant cell seen with a light microscope, and this is the same plant cell that you would see using an electron microscope, and as usual you will see a lot more resolution, a lot more detail with the electron microscope as opposed to the light microscope. So just like the mitochondria, the chloroplast has a double membrane structure. It also contains DNA. So there are three organelles in the plant that contains DNA: the nucleus, the mitochondria, and now we know the chloroplast. Alright? And again, the endosymbiotic theory explains why we believe chloroplast has DNA and why it might have been originally a prokaryotic cell. So you see, well, I already answered this question. State three organelles that have DNA in plant cells. Alright, now if you were listening all this time, you should be able to say which organelles are present in animal cells but not in plant cells. And what are the differences between animal and plant cells? Now, anytime you see a question like this where you have to state differences, I would strongly recommend that you use a table, right? Tabulate the differences. It's going to make life so much easier for you. Alright, so this is just a summary table of all the different organelles and their functions. So you could go back, this could be a nice revision slide here. Alright. Now, some homework I want you all to do some research on. I want you all to tell me the contributions of these scientists towards the cell theory. I want you to state the differences between a light and electron microscope. And lastly, what is the endosymbiotic theory? And give evidence to support it. We have already touched a little on it. I want you to go now and do some more reading on this. Very interesting stuff. So I think you won't have any problem getting the information, and you should enjoy the exercise. Alright? So guys, I really hope that you all learned something today. It was my pleasure to tell you all about a cell. So please keep at it, keep studying. The only way to properly do any science subject is to just keep reading, read different textbooks, get different angles. So this is just a little introduction to the cell, but there's so much more that you could read and learn about. So have fun, and good luck. Okay guys, take care.